So with that, the first speaker is talking on the organism itself, and it's Tamara Doering. And I love this, I get to read about people. Tamara is the alumni endowed professor of molecular biology at Washington University Medical School. Dr. Doering earned her MD and PhD from the Medical Science Training Program at Johns Hopkins Medical School. After postdoctoral training at UC Berkeley with Randy Schekman, and I think got the Nobel Prize, and a visiting scientist position with Arturo Castoval at Albert Einstein College of Medicine. She joined the faculty of Cornell University Medical College, and in 1999, she moved to Washington University, where her research focuses on capsule synthesis, gene regulation, host interactions of Cryptococcus neoformans. She is a fellow of the AAS and the American Academy of Microbiology, a member of the American Society for Clinical Investigation, and a reviewer for NIH grants and scientific programs. So it's my pleasure to introduce Tamara.
Kind of a tall order when you get some applause before you said anything. I could just sit down, but anyway, I won't. So, let's see. All right, so you heard the basics of my background, but since I don't know most of you, I just thought I'd mention how I ended up working on Cryptococcus. Of course, there's many paths to this organism, but I started out actually working on African trypanosomes in a biochemistry department for my PhD work. But this really introduced me to the idea of infectious diseases, and I got very interested in all these wild organisms, particularly those that cause significant problems in areas with limited resources. I then jumped coasts and organisms and started working on the fundamental model yeast, fundamental biology of the model yeast Saccharomyces cerevisiae, but I really missed working on something that directly caused disease, so I combined my interests and ended up working in the middle of the country on Cryptococcus. And what I want to do today is give you just some very short basics of cryptococcal biology. Many of you know this, but just to get us started from the same place. Talking about the tools and approaches we can use to answer questions about this organism, as well as some of the challenges that are inherent in these studies. And I want to stress that while I'll be talking about Cryptococcus as an organism and mostly using capsule biology as an example of the types of problems we can study, much of what I'll say applies to many other organisms and many other scientific questions. And then I'll try to spend a moment at the end just touching on the question that Arturo, for example, often asks about, why is all of this important? So this organism one from a bone infection and one from peach juice. And I kind of like this pair of reports because it reminds us that this is an environmental organism and also something that causes disease. And as you heard last night, this was initially something of a historical curiosity until more recent decades when it obviously, for reasons all of you know well, became a major disease problem. So a few of the key features of the organism that, again, many of you are familiar with is it's a yeast, it's a budding yeast. Here's a parent cell and a bud in an image from Arturo's lab. And it's also distinguished by the polysaccharide capsule, which you can see on the surface in this picture, or in the more classic appearance of how you look at capsule in an India ink stain, where you can see between the cell wall here and the sort of moving particles of ink that constitute this gray background, you can see this halo, which of course corresponds to the space where the capsule fibers are sort of displacing the particles of ink. And of course you can get an idea that it's a quite significant size structure. In terms of some other basics of the organism, you heard from Joe Heitman some of these areas last night. The genome is about 19 megabases on 14 chromosomes, and the annotation suggests something like 7,000 genes for a sort of round number. The organism is usually haploid. It occurs in A and alpha mating types, and these can interact, form these mating structures called basidia. This is a basidiomycete yeast. And they can produce recombinant spores, so can undergo sexual recombination. And there's also a lot of variations on these themes that Joe and others have studied that I won't have time to even summarize. Another really key feature of Cryptococcus neoformans is the research community. So this organism has really been turned into a model pathogen for studies of, pathogenic fungi and the diseases that they cause. And that's been from the concerted effort of a really collaborative and generous group of investigators. And that's really the reason why the field has advanced so quickly over the last couple of decades. And many of those people are here, and you can hear more about this group. So in terms of the type of research areas, and again, I've tried to write these really generally because they can apply to your favorite fungus as well as mine. There's people who are certainly interested in studying explicitly the virulence factors of the organisms, although we can argue over a beverage later what that exactly means. There's others who are interested in unique features of these organisms. And of course, it's of interest to study biology relevant to infection, and that we'll start to touch. You'll hear more about that in the next talk. And these areas, of course, overlap, so there's very few biological features of the organism that fit cleanly into one category. But for example, when we talk about virulence factors, we might think about aspects of the organisms like temperature tolerance. You heard from John Perfect about, or you heard, I guess Arturo mentioned it, but also John has often spoken about this topic. The organism also has the ability to form melanin, as you can see in this upper colony in the picture, and this is a topic that's also been studied by the Casadevall and Williamson's lab and seems to be involved in protecting the organism from environmental stresses. So that's an area of interest. A number of labs study various secreted enzymes, often degradative enzymes that help the organism survive both out in the environment and of course cause problems when that environment is, for example, an infected mammal. And there's also the capsule, which you will hear more about, I promise. In terms of unique features of the organism that are of interest to study, of course, I already alluded to the Heitman's lab beautiful work on various aspects and types of mating and spore formation. There's been a lot of studies also mentioned last night of the genome that has some unique characteristics, and again, the capsule. And finally, just to mention a couple topics in biology relevant to infection, a big area of interest is the response to stress, because of course when this organism enters a mammalian host, that's a big stress. And so there's a number of labs working in this field. And there's also the broader topic of host-cell interactions. So for example, we know that cryptococcal cells can associate with host phagocytes and be internalized. Usually, of course, that's a good thing. These cells are supposed to get rid of invading microbes, but we also know that this is a facultative intracellular pathogen. So it's quite happy living and proliferating inside a host cell. And there's a lot of ways this can resolve and a lot of interesting biology there that has been studied in the field. And another very interesting problem in interacting with host cells is the question of how these organisms actually get into the brain. So if this is a longitudinal section of a blood vessel in the brain, perhaps a microvessel, you can see that to get into the brain and cause the lethal meningitis that you'll hear more about later, this organism, whether it's free or perhaps inside a host phagocyte, is going to need to cross the blood-brain barrier to get into that space. So all of these are very active areas of research. Now, my focus, as I've mentioned, is the capsule, and I'm going to use it as examples this morning because I know the most about it. It's what most of the work in my lab has been on, especially until recently. So I'm just going to use it to inform the discussion, but again, this can be widely applied. So how can we study the capsule? The first thing we can do is what you've been doing so far this morning since breakfast, which is just looking. So there's lots of ways to image capsule. We can look at the light level, and here's another example of an India ink stain, like I mentioned, sort of classic way to identify this organism and visualize its capsule. We can also use reagents that are more specific, like antibodies. These are monoclonal antibodies from Arturo Casadevall, that in this case we're using to stain the surface of a cryptococcal cell, in this particular experiment differentially staining the parent cell and the bud. So we can just look by light microscopy. If we want higher resolution, we can look by electron microscopy. So these two images were from methods that are optimized to look at different things. On the left, you're looking at a section that was prepared in a way to really highlight cell membranes. So you can beautifully see the nuclear membrane, even the pores. You can see mitochondria and secretory vesicles. On the right, the image was prepared so that we could really see carbohydrate structures better. So now you can see the sort of two layers of the cell wall and the capsule very beautifully. But obviously, there's a lot you can do and a lot you can learn using these imaging methods. Other modalities of electron microscopy give you more of a 3D picture of the fungus. So here you can see in a scanning EM how really beautiful these cells look and their globular nature. Or you can crack open cells and use other images or other types of imaging like this quick freeze deep etch picture where you're looking right up at the edge of the cell. So this is actually a bit of plasma membrane. Those dots are proteins, which I find astounding. And then here you see the cell wall coming around. There's a brand new bud forming. You can again see it's sort of two-layered wall. And then coming out from this wall are these beautiful, intricate array of fibers. And this is the capsule that has really caught and held my attention for many years. We also, of course, want to image the interactions of these cells, of the fungal cells with host cells. And of course, you can do that also. You can do it at the light level. For example, here are mouse and human cell lines. These are phagocytic cells, and you can see how avidly they've taken up the cryptococcal cells inside. We can use confocal imaging. Here's an example where we've stained the cell wall of the fungus and the cytosol of the host. And this enables you now to determine, for example, whether these fungi are just associated with the outside of the cell or whether they've actually been internalized to get at some of those cell type interaction questions that I mentioned. And of course, we can do histology, and many of you are familiar with images like this, although maybe not quite this bad. This is actually a section of mouse lung, and you can see the mucicarmine stained budding yeasts and even the space occupied by their capsules. Another way to image with host cells and really highlight the fungal organism is to take advantage of the molecular techniques that I'll mention in a minute to have the fungal cells express some kind of fluorescent molecule. So here you can see the yeasts are actually expressing a red fluorophore. These are cells that from Jenny Lodge's lab. And this is a really nice way to highlight them then interacting with host cells. And in this particular image, what you're looking at is phagocytes in bright green that have engulfed these cryptococcal cells, although some have escaped. And they're actually on a background of endothelial cells here, because as I said, we and others are interested in the interactions of these cells with brain, in particular with brain endothelium. And finally, there's been advances particularly in the last decade or so, that have made automated imaging really accessible on the level of individual labs. And the type of thing I mean is not only instruments that will automatically take many, many images, so you don't have to have someone, sitting at the microscope as long, but then there's software that will annotate the images and enumerate the various constituents of those, or the various features of those images. So for example, here's a picture like what I showed you before of fungal cells interacting with host cells. The host cells in pink and the fungi in green. And here, the sort of automated software is identifying which of these host cells are actually interacting with fungi, either adherent or internalized. And on the right are a couple of cryptococcal cells, now just looking at the cells themselves that are stained on the cell wall and capsule. And now again, the automated images are automatically annotated to identify where the edge of each of those structures are if we want to, for example, be able to very quickly assess capsule sizes. So I'll just wind up on imaging to say that, of course, these are lots of beautiful pictures, which hopefully was a nice way to start your scientific day. But there's certainly challenges with all of these methods, or to various extents. One problem, especially if you're interested in carbohydrates, as many people who study fungi are, is imaging proteins and glycans together, because they're very different types of molecules, so optimizing each can be difficult. The wall and capsule present a challenge if you want to get molecules like antibodies inside to stain cells. And there's been some issues of sort of just expressing fluorescent proteins, although people have been having more success with that lately. So another area where we have, but obviously these are powerful tools. And another area where we have really powerful tools is sort of molecular tools. So what can we do with a cryptococcal cell? We can delete genes, we can insert genes, that's at the chromosomal level. We can express genes on plasmids, so that would be sort of episomal expression, and both of these are sort of enabled by a number of selective markers that have been developed by different labs over the years, and even methods to reuse markers. And there's various promoters that allow you to have either regulated gene expression or constitutive gene expression. And then beyond these ways of manipulating gene expression in the genome, there's also some other techniques. So we can use RNA interference to specifically, or reasonably specifically, reduce expression of genes. And as was mentioned last night, there's a very recent report using CRISPR in this system. So just for an example of the way we can use these kind of tools to test hypotheses, quite a number of years ago, we were interested in what was really happening at this boundary where the capsule fibers that I showed you are interacting with the cell wall. And we had some evidence that perhaps one of the cell wall components, alpha-glucan, was involved in this association. So we could use these molecular tools to test that idea. And for these experiments, we used actually another type of imaging, which was immuno-EM, where we were interested in alpha-glucan. We had an antibody to it that was linked to a gold beads, and they show up in the EM as these little black dots. So you can localize that molecule in the cell wall. You can see the capsule fibers here. And then when we delete the gene that makes that alpha-glucan, you can see that the dots are all gone now, as is the capsule. And we can recover both of them by complementing the gene. So this is just an example of how we can use a molecular tool to test a specific hypothesis and come up with a very conclusive answer. Just an example of one other technique, which is RNA interference. This is another older study where you're looking at wild-type cells up here that are just stained with capsule, like I mentioned, capsule antibody, like I mentioned earlier. There's a mutant in Cryptococcus that results in no capsule production, no staining, and the cells also kind of clump together. And if we use RNA interference to target that gene, that's these bottom panels, you can see the phenotype is exactly the same. So that has been very useful for some experiments also. In terms of the challenges that go along with these kind of molecular studies, one is that this organism really likes to do non-homologous recombination. So if you there's various methods different labs have developed for putting material into chromosomes or into the genome. You always need to go back and make sure there hasn't been any scrambling and things are really where you want them and nowhere else. We also don't have great ways of regulating copy numbers of plasmids, so it means it's a little hard to get exactly the expression levels you want once you start tinkering with the cells. And it would be really handy if we had more tunable systems that could sort of reliably regulate the levels of gene expression. But really, again, there's been tremendous progress. These are relatively small compared to the huge progress that's been made and how useful all of these approaches are. We can also, of course, go a more classical route and take biochemical approaches to studying this organism. And what I mean here is things like protein purification or expression, looking at the activity of different proteins, some structural analysis, although not much has been done in this particular organism. And of course, our question then would be, in the context of capsule, is the biochemistry of capsule synthesis. How is this structure made? And so if we're asking the question of how the capsule is built, I just have to take a second to remind you all about the synthesis of carbohydrate structures, which is that glycan structures are not encoded in the genome. And also, unlike most other macromolecules like proteins or nucleic acids, there's no sort of one polymerizing machinery that's going to generate carbohydrate structures, and they're not linear, they're often branched. So it gets a little complicated, and in fact, the way they're made is by the sequential activity of enzymes called glycosyl transferases or sugar transferases. And what these enzymes do is they take an activated sugar, here the sugar is xylose, denoted by the star, and it's activated as a molecule of UDP xylose. And then they transfer the sugar from this activated molecule to some kind of growing carbohydrate structure, which then becomes modified. And the thing that's important to know about these particular enzymes is they're really specific. So they care about which sugar they're transferring and to what it's being added. They can also be specific for other aspects of the acceptor, as well as, of course, the linkage that's being made. So when you keep all those things in mind, you realize that even to make this relatively simple structure that I've cartooned here, which is a little piece of the major capsule polysaccharide, is going to take quite a number of enzymes. And in fact, there's much more complex polysaccharides made by this organism. So this is a really hard problem to get at the biochemistry from the biochemical end of capsule synthesis. And we've made some progress, sometimes by using other systems. So here's an example where we were interested in an enzyme that makes that UDP xylose donor. It makes it from a different donor that's also required for the capsule. And this was a case where we had a hint as to what the gene was that might encode a protein that could do this. So we were able to express that gene in bacterial cells here and purify it, and then use a chromatographic assay. The details don't matter here, but just to show that the first precursor, this one, was converted to the second only when that protein was present. So we have good success, often. Now this was in bacteria, so the biochemistry was a little more straightforward. It's actually harder when you're starting with the fungal cell, and I think, again, this applies to many fungal cells. Breaking them open is hard. In our case, we get the contamination with capsule, if you're studying glycoactive enzymes can be complicated. And there's some inherent challenges in glycobiology that we could perhaps discuss later as well. So what other approaches we can take? We can look from through the lens of cell biology, where you're working at the whole cell level, or perhaps thinking about organelle biology. And in this case, often model yeast can help, although certainly only up to a point. And just to give you an example of a sort of cell biological question, we might ask, where is capsule made, rather than the biochemical question of how is capsule made. And this is a question we thought about a number of years ago because, as many of you know, there's molecules, carbohydrate molecules, such as those involved in the cell wall that can be made at the cell surface, and then there's a lot of carbohydrate or glycans are made inside the cell in the secretory pathway. And we actually had some data to support the latter idea, which is that they were made inside the cell. But then you have the problem of how does it get out, because the capsule at the end has to be displayed on the surface of the cell. And so we speculated that maybe the way it got out was via the secretory pathway. And many of you have know that proteins use a secretory pathway to get out of the cell. They're made at the endoplasmic reticulum, taking you back to some cell biology course. They're made at the endoplasmic reticulum. They can be packaged into vesicles that move to the Golgi apparatus for modification, and then out to the surface of the cell, where they fuse with the plasma membrane and release their contents. So that's what proteins can do. And so we thought maybe capsule was using the same mechanism. And we had the idea that perhaps if we could block that end step of this pathway, this is analogous to studies that have been done in other yeast, we could block secretion and accumulate these secretory vesicles and then just ask if the capsule polysaccharides are in there. And so we were able to do this, and here you see the mutant in Cryptococcus with all of the sort of accumulated secretory vesicles. And then we could use that immuno-EM technique that I told you before to look for capsule materials, and we find it indeed in these vesicles. So again, this is just an example of how we can use cell biological approaches to study capsule synthesis. And of course, there's challenges here as well. This is, at least in Cryptococcus, there's been a lot less work in this area, and so that just means fewer people working on it. We don't have great methods for fractionating cells. We don't have great markers. And there's also some challenges with imaging just because of the nature of the cells. So this is an example where we had ways of identifying in a light micrograph two proteins that both occur in the Golgi. It doesn't matter what they are. One was labeled green and one red. So you can tell that they overlap because you see nice yellow dots around this blue nucleus. But at the end of the day, these are big dots in a small cell. So it doesn't really give you a ton of information, although newer microscopy techniques that have just come on the horizon or come up in the last few years may certainly help with this. Okay, a huge area of really useful approaches is genetic ones. And I already mentioned earlier that we can use specific molecular manipulations, for example, to test hypotheses. But another really powerful approach is to do genetic screens. And here what you would do is first mutagenize the organism, and this has been done in a couple of ways. Several labs have used chemical or biological approaches to do random mutagenesis on a population of cryptococcal cells, and then they can look for features of interest or behaviors of interest or defects of interest in those cells. And also, as was mentioned last night, Hitan Madani's lab has been constructing a library of deletion mutants in Cryptococcus. And in a recent communication to the community, he mentioned that they're now well up over 4,000 mutants. And this is really amazing. Remember, I said there's about 7,000 genes, and some of those are going to be essential, so you can't knock them out. So this is really tremendous progress and a really powerful resource for the community. I'll just give you a quick example of one screen that I've done, although, again, many of people in the field and people here have done this sort of analysis. This is a screen going back to those interactions of the host cells and the fungi. We were kind of interested in which fungal genes would influence this interaction. And so we used our sort of automated microscopy and annotation to assess that for the 1,200 screens that the 1,200 genes mutants that Hitan's lab had initially made. And so you get a frequency distribution here over, this is essentially how avidly these cells are taken up by the phagocytes. And you can see there's some outliers, which might be of interest to us. And we were particularly interested in a couple of the mutants that seemed to have higher than normal uptake by these host cells. A couple of these were genes that had been studied by Andy Alspau's lab, and we took a look at another one. And I'll just show you a couple of phenotypes of this hit to show you that we can get things out of this type of analysis where, in our case, we're letting the phagocytic cell tell us what's important about the fungus. We're letting it lead us to the interesting biology. So this particular mutant, if you look at serial dilutions, this is wild type, and it's on medium where it can melanize, and you can see that these colonies are much paler. This is a complement and another control. And the reason they're paler is they're releasing too much melanin into the medium, as you can see in this bottom image. Another really amazing phenotype of these cells is their shape. So here's wild type cells that I already showed you, whereas on the bottom is this particular mutant, and you can see it's got this crazy sort of crumpled, crushed in, collapsed morphology. And what's really surprising to me is these grow at a pretty normal rate, but they obviously would have a lot of trouble, or they do have a lot of trouble, for example, in an animal. And this strain is also a virulent in animal models of infection, which I'm not going to discuss in this talk. And we've been able to figure out actually the biochemical mechanism that explains all of the phenotypes and the avirulence of this organism, and I don't have time to go through that. So the last topic that I wanted to cover, just winding up here, is the whole, actually, by a variety of techniques. You could look at proteins, you can look at metabolites, or for example, lipids, which have been looked at in crypto. Or you can look, my interest, I guess, at the glycans, the carbohydrate structures that are made by this organism. But you don't want to just collect all this information in a descriptive way. What you really want to be able to do is use it for something which usually requires computational modeling just because of the size of the data sets. And what you want to be able to do is model the behavior of the complex system and ideally make testable predictions using your models. And so where we've tried to do this type of analysis is, of course, coming back to Capsule on the question of how capsule size responds to the environment, because we know that the same strain of cells grown in two different medium conditions can go from having essentially no visible capsule on India ink to having a quite large capsule. And we know this is physiologically relevant because in animals, these cells make large capsules. So what we did initially to look at this from a sort of systematic approach was to grow cells in a lot of conditions that made capsule sizes somewhere between these two extremes. And then we looked at gene expression to ask if maybe there was a set of genes that sort of went up, or sort of a signature of capsule expression or capsule synthesis. And we actually found some genes like that. So what you're looking at here is a heat map of gene expression. Each column is cells grown in a different condition that gives rise to these capsule sizes on top, so they're arranged in order. And what you can see is that we found genes whose expression went up, sort of toward the yellow high expression, as capsule went up, and other genes that behaved in the opposite way. And we were excited about these genes because a number of them had already been implicated in capsule's function or regulation in some way. And also a bunch of them looked like DNA binding proteins, and what I forgot to say is that there's, of course, many levels that you might be active in capsule regulation, and we decided to start with transcriptional regulation, so gene expression. So anyway, we were able to test a couple of our ideas about a couple of the DNA binding proteins by deleting them. As I've told you before, that's a nice way to test hypotheses. And some of these ones that went up in expression as capsule went up, when you knock them out, the capsule is small, which you might expect. And the reverse for some of the members of this other group that we tested. So this was really great and a way to get at some interesting genes that we went on to study. But again, this is a little more of a descriptive approach. And what I've told you is we want to try to look really big picture and do sort of modeling of our system. And so I'm not going to take the time to go over that, but I'll just say that we've been working with computational biologists to sort of map out the transcriptional network of Cryptococcus. So how do the different transcription factors interact with their various targets? And this has been a really great way to make various predictions. So one thing we were able to do is sort of predict which transcription factors might be involved in capsule. And this is before the work you heard last night about one approach is to knock out all the transcription factors and look, but what we wanted to know is whether our model could lead us to the same information more efficiently. And in fact, it did. We were able to predict a bunch of genes that would be involved in capsule, and when we deleted them, you see changes in capsule, even just looking at size. So here's wild type, and you can see some have small capsules and some have big capsules. So this was able, sort of the modeling could lead us to the biology that we wanted to study. We've also been able to use this modeling approach to look at the interactions between different regulatory factors, to identify the binding sites of different factors, and then look at their targets and get to mechanism. And overall, it's just been a very productive way to analyze cryptococcal biology that highlights the power of working together with computational approaches and experimental follow-up. And it's also pointed us, just the last slide, it's also pointed us to some really interesting individual regulators. So for example, this is one where when you delete it, the capsule gets particularly large. I'm not showing you the control, it goes back. Another interesting thing about this particular mutant is that when you infect mice intranasally and look at their survival over time, you can see that the mice survive much longer than all of the wild type and controls. And something that really intrigued us about the way these mice die is that while these animals die with sort of central nervous system symptoms, they're having meningitis, that's what's killing them. These particular animals actually die of pneumonia, and this is the slide that I showed you, or the image that I showed you earlier. So again, this is not a gene we would have knocked out if our model hadn't pointed us in this direction. So just to close, why is this important? I think happily in this group I probably don't have to convince you, I hope I don't have to convince you too hard of the importance of studying the fundamental biology of these organisms. There's basic science value. It can inform other pathogens, other yeasts, other eukaryotic pathogens, and probably beyond as well. And then we can kind of go in both directions. We can elucidate broader biology, like that UDP xylose synthesis enzyme that I showed you. It's also very important in plants and in mammals. It was kind of fun that the first place it was worked out was in Cryptococcus. And then, of course, we're also all very interested in the unique aspects of this organism's biology. And I'll just close by acknowledging the current members of my lab, who you see their smiling faces, but in fact, a lot of what I showed you was done by former lab members, because I was trying to sort of get a broad range of experiments, and also acknowledge our computational collaborator, Michael Brent. And I guess we're deferring questions till later, so thank you for your attention.